Thank you. What I'm going to talk about today, that there is a cycle which is called methionine cycle, which actually add a molecule of methyl group to protein, to DNA, to phospholipids and all others in order to activate, to get the thing going in the cell, to regenerate the process. And what we find out now more and more, 5 years or 10 years ago rather than now, is more and more having some mutation, a single chain, which is like a one single molecule of either purine or pyrimidine has been changed into another one chemically due to some epigenetic syndrome or many other syndrome, and also inherited. So there are many of them, and at least in the methionine cycle we found out there are 10 different enzymes which runs the entire methionine cycle. Each one have of them some kind of a problem with the mutation. So if I just introduce you the cycle, which is extremely complicated now, but once we explain the one cycle which actually converts methionine to the chain of reaction to homocysteine and then goes back to the methionine. That's one cycle. In order to run this cycle, the homocysteine either goes to the chain of events to making glutathione, or then goes to the methionine depending on the condition, whether the person is in oxidative stress or reducive stress. Now, in order to regenerate this methionine B12, the enzyme which is required for this function is called methionine synthase, which actually requires 5-methyl tetrahydrofolate, which we just heard from Dr. Mian. It's the most active principle of folate, and then it actually goes back to the tetrahydrofolate, then it comes back and regenerate the whole system of the methionine cycle. So the whole thing runs very nicely if everything goes all right, but when the mutation takes place, then there is a problem. Then on the third one, in order to regenerate that, there is one active group here, 10,4-mil tetrahydrofolate. We found that this is one of the most notorious product actually in the body if it produces in a large quantity, because in particular in case of cancer, this 10,4-mil tetrahydrofolate goes way up, and because this is a precursor of purine biosynthesis, actually helps the DNA to grow much rapidly, and which DNA is a cancerous DNA. So that helps the cancer cell goes much faster. Then, as I said, if the reducing condition, which is an ideal condition, the tetrahydrofolate can come and then regenerate the whole thing. But in oxidative condition, instead of coming this way, it flows back this way, and that creates life pretty difficult. And if we are involved with patients with different kinds of cancer, and number 2 is chronic fatigue syndrome, people suffering with a consistent and long-term problem with the allergy, autism, and number of other kinds of very difficult disease where we find there are problems with this cycle. Now, as I told you, that first enzyme here, which requires methionine to convert into S-adenosylmethionine, which actually helps you to make the DNA methylation, that is one enzyme which is called methionine, the enzyme which actually converts with the help of ATP into that. Now, here we found there is a partial block in some cases, particularly in case of chronic fatigue syndrome. The people do not make enough of SAM because of that block. Then SAM becomes SA, which is a methyl transferase. There are 2 enzymes involved in that, and we also have some people who has a problem with this. Then SA, which actually is a product of the SAM, which actually does a inhibition, product inhibition to this enzyme. So, if you build up this molecule, you will have inhibition of this reaction. So, it's a very toxic molecule, has to get rid of as soon as possible. Now, the way to get rid of these, there is an enzyme called hydrolase, which converts her into adenosine and homocysteine. In a normal condition, no problem, it comes. In case of severe allergy, this is where there's a major trouble starts. If we don't give support to the patient, some immunological support, and then the patient will not be able to convert and it will back up, it will block this reaction. So, that's a very vicious cycle they created, and whatever homocysteine is produced, you have to make some glutathione, which is antioxidative, and it actually helps you detoxify. It actually reduces a lot of oxidative product. So, you cannot make this very healthy product to have in the body, and that you cannot make if there is a problem here. And then of course, the cycle breaks down completely. So, I will go over each step about. Most vulnerable one is this one, MTHFR. That one is the one almost, there are 40 mutations in that small 40,000 molecular weight protein. But out of them, 2 of them are the most common and most serious. One is called 677, which is a name where they go, and one is 1298. Those 2 are the most common one. But the other 38 of them and more are new coming, is this one gets mutation for all kinds of. If you take a methotrexate, this causes problem here. If you take some other 5-fluorouracil, it causes problem there. So, lot of drug causes problem, and it is also inherited from the mother. Now, there are 2 types of molecules you make. One is homozygous and another is heterozygous. I will go over that later on in little more detail. The MTHFR, if one chain is a normal chain and one chain is a mutated, it's called heterozygous. So, one mutated chain, and DNA is a double chain, right? So, one chain is a normal and the other one is a mutated one. Then it's heterozygous. It's a less of a problem to cure. I mean, make it better. But if it's homozygous, so both are changed, then it is pretty bad. Now, on the top of that, the other one is 1298 product of this mutation. That has also a severe issue with the sulfur metabolism. And then it actually messes up all the other this particular chain of reaction completely. So, and if you have one chain of the 677 and another chain is 1298, then it's a disaster. I never have seen anybody who has that kind of situation has recovered. So, anyway, I will go over all the steps one at a time and to tell you that what you can do about it later on. But in the beginning, I will just introduce you to those enzymes. Okay? Now, beside this methionine cycle, there is 2 other enzymes we will be talking about, because they directly interlinked with this methionine cycle. One is COMT, it's a catecholamine O methyl transferase, which helps breaking down the neurotransmitter. So, the methionine synthase and the methionine cycle makes a lot of methylation, makes a lot of neurotransmitter. For example, norepinephrine to epinephrine is a methylation product, estrogen methylation, but this enzyme actually helps breaking down. So, 2 enzymes work side by side with 2 opposite consequences, and if this one is mutated, and which happens to very commonly with the people first particularly from originally from China, Taiwan, this kind of these people have this mutation pretty common, and the MAO, which also help breaking down the neurotransmitter, that also if it's a mutation, along with the mutation, for example, MTHFR, there is a very disastrous situation. Very hard to do any manipulation with them. Now, what I wanted to emphasize that with the last 5 years of work with somewhere around 80 physicians specialized in this system and about 5 scientists, we developed some technique which you can use to make the life of these people who has a mutation lot better, if not completely better. And this is you can do simply by manipulating different types of supplements. Okay. Now, Dr. Mian just explained about the folic acid. We have a little more add to the thing which he's mentioned to you. Folic acid is made of 3 different units. This unit called pteridine unit, this called para-aminobenzoic acid, and this unit is called glutamic acid. All 3 are joined together by covalent linkage to make folic acid. When you buy synthetic folic acid from the market, that's what you get, this, and 1 residue of glutamate. Now, for biological purpose, the natural product like spinach or any other dark green vegetables, this glutamic acid is actually 6 residues. So, natural one has 6 residue glutamic acid and the synthetic one has only 1 residue of glutamic acid. Now, in some cases 1 residue works out, but in some cases, in methylation case, when there is a damage, then you definitely need 6 residues. And there are enzymes which actually, when you look at the, if you look at the panel we do, where we try to measure the folic acid in the red blood cells, and we also try to measure folic acid in the serum. And we can show you they make a very big difference. And then folic acid, when you say, is just these, but when you say folates, that is a mixture of methyl tetrahydrofolate, 5-formyl tetrahydrofolate, 10-formyl tetrahydrofolate, all these are together is called folate. So, total reserve in the inside the RBC, that's what we are going to talk about. So, now okay. Now, simple thing is folic acid when you take, then it goes into the dihydrofolate with the help of NADPH. Now, this enzyme called folate reductase, and this is in a human is a very limited supply. So, if you take a huge amount of folic acid, it does not help at all because it remains as a folic acid, it does not go into chain of events to make 5-methyl tetrahydrofolate. So, what happens, in fact, it does some harm. If you give a huge amount of folic acid and it does not convert into 5-methyl tetrahydrofolate or anywhere in between, then actually the surplus folic acid has a lot of side reactions. So, and then it regenerates itself, there is no problem with that, and this is 5-formyl tetrahydrofolate, it comes one way to make 10-formyl and then it makes into 5-methyl tetrahydrofolate. So, from folic acid, it goes all this chain of reaction to make 5-methyl tetrahydrofolate and then comes back to the original, just to change from homocysteine to methionine in the methionine synthesis. That's the main chain of his, all these steps they have to go through to convert, to close the cycles of methionine. Now, if there are many cases, as we saw in 5-formyl tetrahydrofolate, which is the same thing as a folinic acid, which is a reduced folic acid. Now, what we do, instead of giving folic acid, we give folinic acid to the patient. And we give, and I will talk about the doses a little bit later. Dose makes a big difference, what dose you give to the people who have a mutation. And this one is a particularly important one. We bypass the system, and then of course this one also is a good one, but one has to be, we found very careful when you give 5-methyl tetrahydrofolate. To many patients it can cause even harm if you don't adjust the correct dose. So, Important point here is two things. One is the dose, another one which form you give to the patient. Now, in order to maintain a proper quantity of folic acid in the cell has to make it 6 residue of glutamic acid, because when this converts into this chain of sequence, the enzyme works much better when the there is 6 residue in the folic acid. But when you give only 1 residue of folic acid, it actually blocks down around here. So, and also rest of it is just passes away through the body. And that's another important point of this folic acid treatment. Okay. Now I switch over to methionine synthase, the enzyme which actually makes from tetrahydrofolate to methionine. That has a 3 active site: SAM, homocysteine, and 5-methyl tetrahydrofolate. And then the active site of the vitamin B12 is cobalt, which is a trivalent, which is a basic activity, which I will show you later how it becomes that. But the reaction of the methionine synthase, we found the blockage is in here and here. As I said, that you have to know where, how to give and how much to give. If you know where the blockage is, you can easily adjust. Here, the person who has a blockage here, the mutation here, and you give a large dose of this, the patient becomes very jittery, very sick sometimes, sick for many days, unless or until you withdraw the product for them. So my suggestion will be at the end that you have to know exactly what dose you are going to give, or how to adjust. Different people has a different tolerance, so you may have to adjust the dose titrate the dose for persons whom you are working with. Okay, then methylcobalamin is biosynthesis is very important. There are hydroxycobalamin. There are 4 forms of cobalamin you can get. 3 are natural. 1 is a synthetic. Cyanocobalamin actually is a vitamin B12. When you isolate and purify it, during the process of purification, it becomes a cyano group attached to the active site. So that is artificially made cyanocobalamin. The other ones is a hydroxycobalamin. When it goes with the glutathione, that's why I told glutathione is so important, become the glutathioneal cobalamin, or cyanocobalamin with the glutathione, it becomes the glutathioneal cobalamin. That's with the help of SAM, it becomes the methylcobalamin. Now, that is why I said if there is a blockage in making SAM, you cannot make methylcobalamin. And if you don't make methylcobalamin, you cannot convert homocysteine to methionine. So you break the cycle altogether. So the important point here is that the SAM, which actually helps you, if there's a blockage there, you have to support this enzyme, and then only you can get about 40 to 80, depending on the person, depending on the place where actually mutation took place, you can activity back to that extent. Okay, and now what happens in the glutathione cycle? This is another one which we'll be talking. I'm just introducing it now for you to. Glutathione continuously become reduced and oxidized. This is the one which you need, reduced product. Now, it oxidizes to oxidize glutathione with the help of an enzyme called glutathione peroxidase. And then it reduces down again, the cycle continuously going on with the glutathione reductase. And this enzyme requires vitamin B2. So if anybody who has a low level of reduced glutathione and high level of oxidized glutathione, if you give them an appropriate dose of vitamin B2, the chances of that you can get the cycle going in a proper way. Okay, another thing I will be talking to is that brain level of GABA, which happens to cancer, chronic fatigue syndrome, and autistic children, particularly in autistic children. We need GABA in our brain. There are lots of GABA in our gut, which is not very helpful because the GABA from our gut does not cross the blood-brain barrier. So when you measure the GABA in the plasma or in the urine, you only measure what is in the gut, not what is in the brain, unless you can measure in the cerebrospinal fluid by spinal tap. So what happened, you take a glutamine or glutamate, it converts into GABA and also alpha-ketoglutarate. In the brain, there are two enzymes called decarboxylase, glutamate decarboxylase, and glutamate transaminase. Now, glutamate transaminase makes alpha-ketoglutarate and decarboxylase makes GABA. Both requires P5P. Now, if there is a shortage of vitamin B6 in the brain, then the primary, whatever is available, transaminase takes over and make alpha-ketoglutarate. They cannot make GABA. And that is a big problem with because the transaminase has about 100-fold more affinity towards the P5P than the decarboxylase. So there are ways to handle that, and I will also talk about that. I'm just introducing some of the new things. When I will be talking mutation, then it will not be very difficult for you to understand. Okay. Now, vitamin B6, I just mentioned, no matter how many ways you give you vitamin B6, with the kinase, it becomes 5-phosphate, and then it becomes P5P, pyridoxal 5-phosphate. Or you get pyridoxamine, and it happens the same way. And then it can convert from pyridoxal to this, and then it can also goes to pyridoxic acid. So all the time, the most vitamin B6 from this end goes two different enzymes and then make P5P. Now you will see at least 30% of our population, particularly mentally retardation, in some chronic fatigue syndrome, and also in autism, there is a gross mutation in this kinase. And we published this paper about 8-9 years ago, that the kinase activity is very low. Actually, there's a SNP. We now have sequenced the whole molecule of the pyridoxal kinase, and there's a SNP there that as a result of that, it cannot make pyridoxal 5-phosphate. Now, you are caught between now rock and a hard place, because when you give P5P instead of pyridoxine, it doesn't help, because P5P cannot cross the biological membrane. Neither it can cross the blood-brain barrier. So when you give P5P, it goes in on the inside, and the problem is that after it passes through duodenum, there's a pool of large amount of lysosomal enzyme, and that enzyme has a considerable amount of phosphatase, and that breaks down P5P to pyridoxine and phosphate. So really it is not helping, but both of them can go inside the cell, phosphorus as well as pyridoxine, and can make P5P inside the cell if the kinase activity and oxidase activity is intact. Now, I don't want to go through these are calculations. I can show, by that I can show you that how much it is effective when there is a, but I will keep that for later at the end of the talk, that this kinase actually is so effective, pyridoxal kinase is unbelievable. Just once we sequenced this one, the genomic sequence, as well as few others, we found out there are 3 vital regions where these are mutated. Okay, this is the thing, is the heart of our talk. Now, how do you know what dose to give which patient? Okay. Now, in order to do, I want to bring just only one case of the pyridoxal kinase, but we have done it the MTHFR, at least 4 other enzymes we have done the similar experiment. What this experiment tells you, that when a normal person, which is a control, takes B6, it reaches around here almost to the Vmax. That means maximum biological activity at this concentration. which is about 0.1, which is a double the RDA. Now, when you give that to this is done on a chronic fatigue syndrome, like a 8 or 10 chronic fatigue patient, and is an average value of that, then you see in order to reach that level, it has to go through all the way to here. So, you need about substantially more amount of vitamin B6 because of the damage in the kinase. Now, when this patient is treated well, you see after treatment, it actually comes to the same value, but it takes, of course, a little longer time, not as good as the normal patient control. So, so what I am trying to emphasize from this slide is that in order to get the maximum activity of the vitamin B6, you have to know if there is a problem with the kinase enzyme, or whether the patient can take and how much he can take. But the easy answer is that you cannot titrate every patient. That's almost very difficult. So what you do, you first give a low dose, and I will come to that, how to administer the dose, and then gradually increase the dose till you find the system is working. So every 3 months we have done this study, and we have shown that you can actually do that by changing the profile. Now, one important thing I want to mention, that we are not changing if the gene has been mutated. There's no way in the world we can change the gene unless the present technology which is coming, which is called the CRISPR technology. I don't know that you're aware of it. What it does is actually edit the gene and repair it when it is necessary. The idea is not very new. It was already known that bacteria does it when they wanted to help themselves from the bacteriophage. So they can actually, there is an enzyme which actually edit the whole DNA and find out where the bad thing has been introduced by the bacteriophage, and they will edit that and then that's how they recover themselves from the phage. So this technology was taken over by the human. First was done by about 5 years ago in rat and mice, and now it is being done in human, and there's a lot of biological ethics committee getting involved in it. But the thing is that you can, in certain cases, can edit a particular gene and insert a new molecule to cure that. But that is, the technology is about 10 to 15 years from now, after all the hurdles, the litigations and everything, when they will do it. But yes, here we are not changing any DNA structure. In the gene, which is in a regular sequence, there are nonsense gene and the regular gene, and in between you can do some manipulation by which you can activate or make the mutated gene work much faster or slower, whichever is necessary. Okay, this is the typical. When you do the pyridoxal kinase in control, you need only 2.84. And in patient, these are 110 patient, all of them have cancer, colorectal, esophageal, something else. Head and neck cancer. These are 3 group of people we used here with 110 of them. See, pyridoxal kinase is mutated, gives 179. So you see, you need that much more excess vitamin B6 in order to do a good job, which it was done before. Same thing, transaminase, decarboxylase. The worst one is the dopamine decarboxylase, which actually is very critical for Parkinson's disease, and you see how much more B6 you need in order to do the proper job. And 5-HT decarboxylase is the same way. You need much more. So the Km, that means Km is a half the substrate concentration. Half of that is actually necessary, and it is very different with a mutated patient. And we have done that about 6 other enzymes. We can show you the same structure. I don't want to show now, but if you want to, if you're interested, I'll be more than happy to send you. If you request us, then I will send you how much more you need of these substrate for those enzymes in order to get this enzyme going. Okay, now we come to the crust of the problem, mutation. This first one is MAT, methionine adenosine synthesis transferase, which takes methionine, ATP and magnesium to make SAM. Okay. There are the mutated one is known as MATA, which cause hypermethylation, and there are two forms, which is a MAT 1A delta one and then a MAT 1A beta. Mutation is affect liver and process methionine slowly. It causes autosomal receptive disorder. Now, there's a lot of things to say about that, what you do and how you help it. What I am trying to tell you is not a very nice thing. And I made this slide with all the recipe, what you do and how much you do and how you titrate. For some obvious reason, I cannot show you that here. What I will do after I show you this, I will show you those slides, but please don't make a copy or photo of it. But if you request us by email, I will be happy to send you as a research product, okay? So that I am not involved. Like recently I have been involved with practicing medicine without a license. That means if I say that if you use vitamin B12 for this disease, I cannot say that. I am not a doctor. So I have really a big problem, and I faced this problem coming back from United States, almost 1 year of litigation with the state. So please bear with me. I will give you those answers. I will show you at the end of the slide, and if you want those, just send me an email. I will be more than happy to send you, saying that for your request I am sending you as a research guide. But you know what I am talking about. Okay. Then these two enzymes, MTR is called methionine synthase and MTRR is methionine reductase. These are both important. They work together, and it converts homocysteine, needs methyl B12 to make methionine, and also causes, if there is a mutation there, the big problem of neural tube defect or final bifid. And this is one, but there are many other diseases I can mention, which I will give you in the other slides at the end. And this is one of the critical one, MTR and MTRR. MTR reductase, all it does is actually bring the cobalt molecule of vitamin B12 in a right form to convert the methionine synthase. And this is the structure. This is the form you need to convert in a cobalt. And after its conversion, it becomes one, and it goes through the whole process, make this form again. So that's the reductase enzyme, and it requires two things: pyridoxine, vitamin B2, and number of other things, and then the activation domain. This is the methylation domain to convert into that, which actually react to make this thing. So the sum together, MTR and MTRR both work in concert with each other. If one of them has a mutation, the other one doesn't function. So it's a very vicious situation. Luckily, not many people have that problem, MTR, MTRR. Okay, this is MTHFR. I have a lot to say about this. Now, unfortunately, a lot of things it's difficult to say, but anyway, I'll tell you that there are two major ones I'm showing you, 677 and, this should be 2198, yeah, those two. There are 40 different mutations in the MTHFR. For autistic children, I find the maximum amount of mutation. And people with a cancer and inherited disease like, there are quite a few, and phenylketonuria and other things, those cases, there are about 20 mutations. But these two are very common. And almost I will say, but having this mutation does not mean that we are all sick. I mean, if I take do all of your sequence of your gene, I can bet any money about 30% of us have one from other mutation of MTHFR, one of these two. But we live perfectly healthy life, we have no problem. Problem is when you are homozygous mutation, that means The DNA, as I mentioned before, has two chains. One chain is a normal chain. The other chain is a mutated 677. That's called heterozygous. That mutation is very, very difficult to cure. I mean, easy to cure. But if you have a homogeneous mutation, that means both chains are mutated, then it's very difficult to handle. Not very difficult, but we can handle that with all the recipe we have developed for the last 5 years. You can really have. It causes the Alzheimer, colon cancer, stroke, depression, recurrent miscarriage. This is the one is the most deadly one, recurrent miscarriage one. And if you see any patient comes in with a recurrent miscarriage complaint, you can definitely go, a couple of things you can do immediately to alleviate their problem, okay? Alleviate the problem. For example, this one is a FAD, that means vitamin B2 dependent enzyme. In many cases, when the mutation causes the uncoupling of vitamin B2 with the enzyme, then you are in a big trouble. So there are ways to handle that too. And this one causes variety of chronic illness, particularly chemical sensitivity, like nickel allergy and all these allergy, this mutation causes migraines, IBS, and as I said in the allergy. So there are two different things happens with these two particular major one. If it is a heterozygous, it is easy to handle. If it is a homozygous, it takes much longer period to handle. Then we get hydrolase. Hydrolase is the enzyme we just mentioned, which breaks down the SAH to adenosine and homocysteine. This enzyme has a copper molecule attached and requires for the activity of the molecule. Now there are many ways this enzyme can go wrong. If somebody has a mutation is responsible for myopathy of muscle, brain, liver, decrease of phosphocoline, choline, albumin, and increased creatine kinase, alanine transfer. So it damages the liver, it damages kidney, it damages a lot of other things. Causes severe allergy throughout their life. No matter what you do, the allergy doesn't seem to go away from them. It just one form or other allergy comes back. It also increases plasma sulfate, ammonia, and hemoglobin. This is one of the deadly one. In normal case, if you have a 40 nanomolar concentration from our lab references of ammonia, is okay. This patient who has the mutation here, there will be 200, quite high, about 3 to 4 times high normal, or even sometimes 10 times high normal of ammonia. BHMT, okay, this enzyme is a bypassing enzyme. I think I forgot to mention that. The nature has created two ways to make the methionine cycle going. If one cycle breaks down, the other cycle tries to react. But it only works in two different tissues, kidney and liver. What happens in the case of this enzyme, it takes the homocysteine and binds with the trimethylglycine, is a TMG, we call trimethylglycine. Together, it makes the homocysteine, see, and make your methionine and DMG. So these are two harmless products. You add them together to you can bypass the system. This system also called cardiac problem, CAD problem, and placenta abruption. That means you actually break down the placenta in the process. So once the placenta is formed, after 2 to 3 weeks within that time, if there is a mutation here, then you know what is the problem. Okay. Now this is an enzyme SHMT, which is serine hydroxy. This is the one which makes this most toxic product, 10-formyl tetrahydrofolate. And toxic for, I mean, this is a chemical substance in a normal body, it doesn't matter, it actually converts. But the serine with the tetrahydrofolate, with the help of P5P, it makes the glycine and tetrahydrofolate. Non-Hodgkin's lymphoma and overexpression of oncogene and inactivation of tumor suppressing gene, low level isolation will produce less glycine, which is agonist of NMDA receptor. So it actually all biochemical process, everything can change with this kind of system, mutation here. And these sulfite oxidase, when actually homocysteine converts into glutathione, there is a pores of sulfur comes out as a free sulfite, and then it also ammonia. Those are the two major one comes out from the system. The reaction takes place in the mitochondria, alters the metabolism of sulfated amino acid, causing seizures and mental retardation. In particularly in mental retardation, this is we found if you give some molybdenum, vitamin B12, and I will mention also what kind of B12 you give, you can actually help this person fairly well. Okay. These are the two enzymes. As I said, there is a mutation here is very common, particularly the people who works in the paint factory, which has a lot of butane and butyl acetate, and those are the people who has this kind of a problem we look, because most of our patients came from that kind of source. And but if it is by itself COMT, it breaks down slowly our neurotransmitter. But if it is with the MTHFR, both are mutation, then of course this because sometimes situation comes which you cannot even help. And one of the major thing here is that schizophrenia and depression, anxiety, bipolar disease, and OCD, that happens from this. And the last one is a MAO. No, I think there's another one, MAO, which actually have the same thing. MAO does epinephrine and norepinephrine to break down. These are the breaking down product of system, so that after the neurotransmitter does its job, it does not bog down into the system and goes on and on and on. It has to break down. So body has a mechanism to do that, but here they cannot do it with that mutation. And the last one is CBS, which actually there are two mutations here, and causes from homocysteine to cystathionine, but then later on it goes down to glutathione process. It's a following component, it produces high level of sulfite, high level of alpha ketoglutarate and glutamate, ammonia, which produces excess cortisol and contributes to adrenal fatigue, and the last one is a low homocysteine. Low means a very low homocysteine. And so I think this is the main mutation thing I have talked to you about, which is very important. And these are just some data which show you that if you have a low cysteine, you can convert into bring it back to the normal. Methyl B12, if you have a low, you can bring it to normal with the treatment. What kind of the treatment we are going to show you. I think these are the things which are very similar. Okay, the folinic acid is very important here. The folinic acid, we use it for various kind of situations, and we use from very little to very high level. And it actually improves SAM, SA, homocysteine, 5-methyl-tetramer, and methionine, all these things. But in this case, of course, we use very high dose, 1 milligram per kilogram body weight, which is extremely high. Normally, you start with the 1 milligram per person and then keep on increasing it. And these are very difficult case. That's why we used it rather high concentration. And you can see that in every case before and after, it has gone up tremendously with the folinic acid. The two reasons for it. One is folinic acid is absorbed much faster. It bypasses the main folate dehydratase system. Third thing, it crosses the blood-brain barrier very quickly. And in CSF, it goes within like a 2 hours time, you can see the reaction of that. But it requires about 3 to 6 months of treatment in order to get any. Okay, I think, yeah, this is one thing I wanted to show you, that in case of cancer patient, particularly here, Nitrotyrosine is a very important factor. Control value is 4.6. In a CFS, you can see 22.7. So the nitrated byproduct, oxidative product, goes pretty high in case of, and 8-hydroxy 2-deoxygasonate, you can go from control 74 to 1.22. So actually, in oxidative stress, you will have this kind of situation. The whole body is exploding with the free radical oxygen. So it is very important sometimes when you see oxidized glutathione, you see high adenosine, you see high SAH. It is very important to see is the person is suffering from nitrated. But this can be really relieved very quickly, very fast, very effectively by proper supplements. Okay, these few slides I will show you very quickly. This was done about 318 patients, and we wanted to make total folate in the RBC. And look at, we started with 330 level, and over 15 months period, you can go almost to 600. So by the appropriate treatment, what we are going to talk about, we really can change the total folate in the RBC, even though there is a gross mutation in different places. This is the folinic acid level. Actually, you can increase from 6 to, this is a CSF patient also, is going from, and these are the, from 318 patients, this is a common average of that. So you can increase the folinic acid by a number of other ways, and also by giving folinic acid. This is the folic acid, plasma folic acid. This is not very useful data, because going out high level of plasma folic acid is not a very good thing. Only thing we wanted to see how many residue of glutamic acid it has. Now these one, the one which had glutamic acid, so we only measured that one. So adenosine level similarly can go down, and the 5-methyl tetrahydrofolate actually can go it starts very slowly in CFS, and then certainly starts pick up. Almost after 9 months of treatment, you get a very little production, but then it goes up. And 10-formyl tetrahydrofolate, which I talked about, pretty it starts from 8 nanomole per liter to all the way to 2 nanomole per liter after 15 months of treatment. So it takes a long time to reduce it, but you can reduce it. In SAMSA, all things can be done after proper nutritional intervention.
So, can you work out the treatment regime simply from the mutations, or do you need to do a really comprehensive analysis of the patient?
If we do just the methylation panel, not the comprehensive analysis, we can take care from there.
So just from the mutation?
Mutation, right, yeah. Second thing, we found as a very interesting thing that if you have a mutation, doesn't mean you have a disease. You still are doing okay. And now there are a number of places you can do it in a very inexpensive way. There are about 20 different genes, you can get a whole sequence done for you. And it costs only $100. 23andMe charges you that. And so you can do the whole analysis done with it. But we never found any effect on the treatment, because on the treatment we have to know what is the product, is short supply or long supply.
Yeah, I guess that's what I mean, that biology has many compensation mechanisms.
Exactly.
So just by looking at a single mutation, you're not going to get an answer.
No. In fact, it has been sometimes misleading in a way. Yeah, MTHFR is one of the worst one as a misleading one.
I just wondered what's in the egg white that is so good for detoxification.
Yes, it can work, and this is partially that one that detoxify. One thing I have found out, without going into very elaborate detoxification like DHMA or DMSA or ADTA. If you simply do the Corella detoxification system, is a kind of Corella means it comes from the moss, the coral. What do you call that? Huh? Algae, yeah. That was very good. Another one I found out also, people have used is charcoal. Takes away a lot of toxins from your body. Charcoal.
What do you think's in the egg white that's doing it?
Oh, there is a called beta hydroxy 3-methyl sulfosalicylate acid. 3-methyl is a beta. It is a complicated. If you want, I'll send you the
Is
structure.
it only in egg white, or do you find it?
Oh
in
no,
other-
there are other places you can find that too. Actually, the most highly content is in egg white. You can get that also in garlic and onion. Those have, That is why sometimes garlic extract has been very helpful in some. And the one is curcumin also helps if you add some bifenthrin. Do you know that about the black pepper? I mix them together. You can get nowadays capsule which is curcumin mixed with, and there is a company which makes that is their only product now, and is with the curcumin and together in a capsule. Any patient who has, we have seen in many like colorectal cancer. If you give in the morning 2 grams capsule of that curcumin with that and some folinic acid and vitamin B6, you see the amazing difference in colorectal cancer. And also subtly it will not metastasize or anything goes around, but it may remain same or better, but I can assure you it actually, we have a very good experience with that, with a lot of doctors.
So what's in the black pepper?
Black pepper actually has a product called allicin, ALLICINE, 3-methyl-allicin, which actually helps curcumin to absorb in your system.
Sorry for that formulation you just mentioned, 2 grams of curcumin and piperine with folinic acid and B6. What exact dosage would you suggest of the folinic acid? And what dosage of folinic acid and B6 would you suggest with the 2 grams?
I would start with this, depending on the age and the gender of the person. I would start with 2 milligram of folinic acid per day and 2 grams of curcumin with bifenthrin. Those are the 2 important ones. What's the third one you're asking? B6. Okay. If you do an analysis of B6, would be very helpful to find out how much B6 he has in the system and how much P5P he has in the system. Because the measurement of B6 contains all these ingredients, and then that way you get a guidance that whether the person can convert B6 to P5P or person cannot do that or partially can do that. But in the beginning, I would start with something like 50 milligram at a time, but then you can continue increasing the amount. 50, yeah. Yes.
Yes, and in our patients, we found that the level of vitamin B12 in the serum, usually it's a normal or high level, despite the patient have got some methylation or mutation of a genetical. How would you explain that?
Okay, there are 2 things. Which B12 we are looking at? Is a hydroxy, methyl, or cyanocobalamin, or what, or a mixture of all. And the most common what it is when you take B12 or take food with the B12, they cannot assimilate. There's a thing called intrinsic factor. And if you have that problem, you can give some intrinsic factor, over 20-25 milligram of intrinsic factor, then you can see the effect. And sometimes we give B12 like a hydroxy B12 or methyl B12, and we don't see any effect, because acrohydium and the stomach is not enough acidic to bind with the intrinsic factor. So you give intrinsic factor with that.
Is it also, I've heard of the problem of unmetabolized folate in the blood. It builds up in the serum, but
sure.
it doesn't go into the cell.
Sure.
Is that possible with B12 as well, that you have high levels in the serum, but it's not going into the cell?
Oh yeah, because there's a molecule called transferrin, which binds with the B12 to bring it inside the thing. There are two types of transferrin 1 and transferrin 2, or 1 and 3. They bind from what happens when you take B12, it goes into your system with the intrinsic factor, then it binds with the, go to the ileum, and then it converts into methyl B12. Then it gets into the cell by a protein called the transferrin, and there are two molecules, two different. Now, if you have, there are people who had, just 1 second, spina bifida. You look at those patients, and they are both transferrin 1 and 2 are mutated. So what you do in that case, to transport the B12 inside the cell, that's the purpose here. And first of all, you can try hydroxy B12, which is much better to get in, okay, rather than methyl B12, and or cyanocobalamin. Cyanocobalamin is the most difficult to get in, because the cyanide groove actually causes a lot of binding problem. So if you give hydroxy or adenosylcobalamin, that also works. It's too expensive, but it also works very much. If you have a problem, anybody with a Alzheimer or something and lack of vitamin B12, adenosylcobalamin goes very quickly to the brain. And actually, in fact, those who have a problem with the memory, like many of us have, and I have the problem, because I sometimes know where I keep the car key every time I forget. And so what you do in that sort of short-term memory case, you take some, at least 500 international unit of hydroxy B12. You'll see the memory improves quite a bit. Short-term memory is difficult to improve. Long-term memory is improving. There are many other formulations you can do to increase the long-term memory. That we have published already in the paper. You can look at it. Yes, sir.
Could I ask a question on absorbed cyanide, please? For many years, I was a specialist in adhesives, and one of the adhesives we used quite a lot of was what I know as cyanoacrylate, and you know as superglue. And I do a lot of repairs. I've got 3 grandchildren, always ask me to repair something, and I find if I leave any of it on the skin, I can feel very strange. But if I scrub it off, is it my imagination, or is the cyanoacrylate? Because obviously there must be cyanide in cyanoacrylate.
Yeah, you can check that very easily. If this patient gets some other product, I can tell you, and it goes down. For example, if you have a situation like that, and if you give folinic acid to this patient and you recover from that system, then your theory is correct. That means the patient has both problem of transporting folinic acid and the B12.
I've never seen anything on a tube of cyanoacrylate that says don't get it on the skin. In fact, I have heard of medical applications where they use cyanoacrylate to join the wound. But if there's cyanide in it and it's absorbed through the skin,
right.
I think you got, because I find if I've been working with it, I have to scrub my hands hard, and then I don't feel, I just feel a bit odd. But when I scrub them, it goes away. So I just felt that there should be some instructions on cyanoacrylate or superglue to say don't let it come in contact, but the medical profession are using it.
Yeah. Do you know another thing they are now using for that sort of situation is a glutathione lotion. That actually helps you also. Is a lotion has some vehicles in it which is added to the formulation, which absorb very quickly glutathione and actually manipulates that situation. So that also, it is one thing to remember. Glutathione, for example, you can give glutathione by injection, which works. It very helpful in detoxification and also making life a little from oxidative stress. But how do you give glutathione? Like autism, in case of autism, you see almost 75% of the kids have low glutathione. Low is very low sometimes, and you can give them glutathione, which is available now orally by liposomal glutathione, which absorbs very slowly and takes a long time. Then you can give by injections, high concentration injection. But recently we found there are some formulations in the market called glutathione lotion. You just add 3 drops this side, and rub your hand like that, or the child, and you will see within 10 minutes it will absorb into the system. And that way you control a lot of seeing, like the Langerhans cells in the skin actually transport that very quickly in your immune system. So it really works very well, the glutathione lotion. And there are many other anecdotal, what we do is, yes?
Well, I was just going to say, I think that we've had the most fantastic afternoon.
Thank you.
We've been given the most amazing representations from both our colleagues
Thank you.
here. And thank you so much.
You're very welcome.